Hey everyone, welcome back. So today we're going to talk about smell. So detecting chemicals in the environment is important for organisms to be able to detect food or mates and to be able to avoid dangerous substances, predators, or enemies. Humans have two mechanisms for detecting chemicals in the environment: olfaction and gustation, or smell and taste. So today we're going to talk about taste, and then next time we're going to talk about nope. Today we're going to talk about smell, and then next time we're going to talk about taste. So chemical compounds that cause a sense of odor are called odorants. Odor is a translation of a chemical stimulus to a smell sensation. An odorant is a molecule that is defined by its physiochemical characteristics, which are capable of being translated by the nervous system into the perception of smell. To be smelled, odorants must be volatile, so able to flow through the air, small, and hydrophobic, which means they are repellent to water. However, not all chemical compounds cause a sense of odor, just as not all wavelengths of light can be seen and not all frequencies of sound can be heard. Unlike other senses, smell is tacked onto an organ with other purposes, the nose. The primary purpose of the nose is to filter warm and humidify the air that we breathe. The nose contains small ridges, olfactory cleft, and the olfactory epithelium. The olfactory cleft is a narrow space at the back of the nose into which air flows, where the main olfactory epithelium is. The olfactory epithelium is a secretory mucous membrane in the human nose, which the primary function is to detect odorants that are inhaled in the air. During inhalation, air and chemicals in the air are taken into both nasal passages on the right and on the left. And the ridges of the nose cause turbulence, causing some air to pass through a narrow space called the olfactory cleft in the top of the nasal cavity. Air settles on a mucous membrane or the olfactory epithelium, which contains the smell receptors. The olfactory epithelium is pretty much the retina of the nose. It contains three types of cells: supporting cells, which provide metabolic and physical support for the olfactory sensory neurons; the basal cells, which are the precursor cells to the olfactory sensory neurons; and olfactory sensory neurons, which are the main cell types in the olfactory epithelium. Olfactory sensory neurons are small neurons located beneath a watery mucus layer in the epithelium. The cilia are any of the hair-like protrusions on the dendrites of olfactory sensory neurons. They contain receptor sites for odorant molecules and are the first structures involved in the olfactory signal transduction. Olfactory receptor is the region of the cilia of olfactory sensory neurons where the odorant molecules bind. It takes 7 or 8 odor molecules binding to a receptor to initiate an action potential. It takes about 40 nerve impulses for a smell sensation to be reported. When odorants are detected by receptors in the olfactory sensory neuron, this causes depolarization of the neuron and eventually an action potential that travels to the olfactory bulb. Humans have 20 million olfactory sensory neurons in total between both sides. A bloodhound has about 220 million olfactory sensory neurons. So bloodhounds can detect a much smaller concentration of odorants than humans can. It's been long assumed that human olfaction is quite poor within the animal kingdom. However, more recent evidence suggests that human smell sensitivity is actually pretty good. A re-examination of the literature revealed that the claim that humans were poor smellers was due to pressures from 19th century religion and philosophy, and not actual science. More than likely. So axons of olfactory sensory neurons pass through a bony structure behind the eyebrows to form the olfactory nerve and to connect to the olfactory bulb. This bony structure is a cribriform plate, and it is a structure that is riddled with tiny holes, and it sits at the level of the eyebrows, and it separates the nose from the brain. Olfactory nerves are the first pair of cranial nerves, cranial nerves, and the axons of the olfactory sensory neuron bundle together after passing through the cribriform plate to form the olfactory nerve. Each hemisphere has an olfactory bulb, which in mammals is below the frontal lobe and above the nose. It is a blueberry-sized extension of the brain just above the nose, and this is where scents are first processed. The olfactory bulb processes smell from the ipsilateral nose or nostril, which means basically on the same side of the body. Within the olfactory bulb are spherical structures called the glomeruli, where olfactory sensory neuron axons synapse onto two cell types. The mitral cells, which is the main output neuron for the olfactory bulb, and the tuft cells, which are the secondary output neuron in the olfactory bulb. The glomeruli are the spherical conglomerates containing the incoming axons of the olfactory sensory neurons. Each olfactory sensory neuron converges on two glomeruli, the one that's medial and one that's lateral. The first relay for the olfactory sensory neurons is in this bulb. Patterns of activity in the glomeruli determine which odor is experienced. These are surrounded by several layers of cells: the juxtaglomerular neurons, the tuft cells, the mitral cells, and the granule cells. So, olfactory bulbs send axons to several other brain structures: the piriform cortex, which is the primary olfactory cortex, the amygdala hippocampal complex, which are basically structures in the limbic system that are important for emotional and episodic memory, the entorhinal cortex, which provides major sensory input to the hippocampus. And it's important to note that there is no thalamic nucleus for smell. And that's likely because smell is so important that we need super fast processing. So if you smell something that is likely to be a poison, you should probably stop smelling it right away and leave. Or if you smell smoke, you should get out, all those important things. So it's probably very important evolutionarily to have no thalamic pathway. The primary olfactory cortex is the neural area where the olfactory information is first processed. And this includes the amygdala, parahippocampal gyrus, and interconnected areas, and also the entorhinal cortex. So the limbic system is the encompassing group of neural structures that include the olfactory cortex, the amygdala, hippocampus, piriform cortex, and the entorhinal cortex. It is involved in many aspects of emotion and memory. Olfaction is unique among the senses for its direct and intimate connection to the limbic system. Olfaction's unique connection to the limbic system explains why scents tend to have such strong emotional associations. So Buck and Axel in 1991 found that the genome contains about 1,000 different olfactory receptor genes. Each codes for a single type of olfactory receptor. All mammals have pretty much the same set of 1,000 genes. However, some genes are non-functional, and those non-functional genes are called pseudogenes. Animals with more functional genes have a better sense of smell. So in dogs and mice, about 20% of those receptors those genes are pseudogenes. which means about 80% are functional. In humans, though, between 60 and 70% are pseudogenes. Each person has a different number of pseudogenes, resulting in individual differences in sensitivity to smells. There may be an evolutionary trade-off between vision and olfaction then. Odorants can stimulate somatosensory system through the polymodal nociceptors, so touch, pain, and temperature receptors. This is responsible for the cool sensations of menthol, the tearing from onions, and the burning from chilies. Also, the sneezing when you sniff black or white pepper. These sensations are mediated by the trigeminal nerve, or the fifth cranial nerve. Often it's impossible to distinguish between sensations traveling up the cranial nerve 1 from olfactory receptors and those traveling up cranial nerve 5 from somatosensory receptors. So the shape pattern theory is a current dominant biochemical theory for how chemicals come to be perceived as specific odors. It states that different receptors detect different odorants because the molecules have a shape that fit well with a particular receptor shape. These various arrays produce specific firing patterns of neurons in the olfactory bulb, which then determine the particular sense we perceive. The vibration theory proposes that every perceived smell has a different vibrational frequency, and that molecules that produce the same vibrational frequencies will smell the same, even if they have different molecular structure. However, other researchers have failed to support this theory. So isomers are different forms of the same molecule in which the spatial arrangements of the atoms are mirror image rotations of one another, so like the right and the left hand. Such molecules are perceived differently, possibly because they don't fit onto the same receptors. So, for example, the D-carvone smell like caraway, and the L-carvone smells like spearmint. So, two very, very different scents, and though these are basically the same molecule, but they're isomers. So, vibration theory then cannot account for this difference in smell because these molecules will vibrate in the same manner. So, the space pattern theory can, which is why it's a leading theory. Most smells we encounter are mixtures of multiple chemicals. We rarely smell pure odorants. People have difficulty detecting individual components of smells and instead perceive like a unitary percept like in other sensory modalities. So how do we process the components in an odorant mixture? So the answer to this is through analysis and synthesis. An example of analysis from an auditory mixture example would be when the high note and the low note can be played together, but we can detect each as an individual note. And then an example of synthesis that we'll take from color mixtures would be like mixing red and green. And this results in a yellow light, but we can't separately perceive the red and the green in the yellow. Olfaction is primarily a synthetic sense, but there's some analytical abilities that can be developed for this. So when first exposed to a smell, it's very noticeable, but over time, the smell becomes less salient, and this is due to an adaptation process in the olfactory receptors. For example, when you walk into a bakery, you can smell the fresh bread for a few minutes, but that typically fades over time. Another example is someone who wears perfume every day. They tend to not be able to smell it and then tend to over scent themselves. So they wear way too much perfume and they can't smell it, but it's somewhat offensive to folks within their general vicinity. On average, it takes about 15 to 20 minutes for olfactory receptors to stop responding to an odorant after prolonged exposure. So receptor recycling happens when an odorant binds to receptors. The receptor then buries itself in the cell body and is therefore no longer available to receive odorants until it reemerges in a number of minutes. During this process, a number of cellular events occur, resulting in the opening of sodium channels. Cross-adaptation is a reduction in detection of an odorant following exposure to another odorant. And this is presumed to occur because the two odorants share one or more olfactory receptors for their transduction, but the other order of odorants also plays a role. Cognitive habituation is a psychological process by which, after a long-term exposure to an odorant, one is no longer able to detect that odorant or has a very diminished detection ability. An example of this would be going out of town and then coming back into town and noticing how your house smells. So when frequently exposed to an odorant, people don't perceive it. So the smell of your house or maybe a chemical in the workplace. Unlike normal sensory adaptation, this habituation takes weeks to reverse. There are three mechanisms that may be involved here. The olfactory receptors internalized into cell bodies during odor adaptation may be hindered after continuous exposure, and this then makes them take longer to recycle. Or the odorant molecules, the odorant molecules may be absorbed into the bloodstream, causing adaptation to continue. As well as some cognitive emotional factors, like the increased sensitivity to a smell when it's labeled as harmful, compared to the same smell if it's labeled as healthful. This may seem like a minor point, but it has real-world consequences for things like smelling smoke from a fire in your home. This underscores the importance of attention and conscious perception. We cannot smell while we're asleep. So attention increases our ability to detect odor, but attention is cut off during sleep, and then so is our ability to respond to odors, which is why making sure your smoke detectors have fresh batteries, so you will wake up when that goes off because you're less likely to smell the smoke and wake up. So the liking dimension of odor perception is typically measured with scales pertaining to an odorant's perceived pleasantness, familiarity, and intensity. We tend to like odors we have smelled many times before. Smells that are considered pleasant have an inverted U-shape function for the intensity and pleasantness. Intensity has a more complicated relationship with odor liking. Smells that are considered unpleasant decrease in pleasantness with more intensity. Hedonic responses to odors being innate or learned has been a subject of debate. There's evidence from infants that odor preferences are often very different from adults, so this suggests some kind of learning association. And then cross-cultural data also supports associated learning. So the evolutionary argument is that some animals exhibit an instinctive aversion to smells from predators. And we also know that you also can learn aversions, like in the learned taste aversion. And so here the avoidance of a novel flavor, or this refers to the avoidance of a novel flavor after it's been paired with gastric illness. So this fast type of conditioning might be evolutionarily supportive for learning to not ingest poisons. But also it shows that you do learn how to avoid things associated with that. So there are two caveats for the theory that odor hedonics are mostly learned. Trigeminally irritating odors may elicit pain responses, and all humans have an innate drive to avoid pain. There is also potential variability in receptor genes and pseudogenes that are expressed across individuals. So let's talk a little bit about what happens when things go wrong with smelling. So anosmia is a total inability to smell, and it often results from something like a sinus illness or a head trauma. So a hard blow to the front of the head can cause the cribriform plate to be jarred back or fractured, and this then slices off the fragile olfactory neurons. So if you've ever done a brain dissection or looked at a picture of a brain online, you'll see the olfactory bulbs kind of hang off by little threads. So they're very easy to harm if you hit the front of your head really hard, like up by your nose or eyebrows. So anosmia causes a profound loss of taste as well as smell. And it's estimated that as many as 1 in 20 suffer from a reduced sense of smell. Specific anosmia is the inability to smell one specific compound with otherwise normal smell perception. So about 20 to 40% of the population have specific anosmia to androstenone, which is a molecule found in armpits, sweat, and pork. Of those who can smell it, most perceive it as an unpleasant, like urinous odor, and the remainder perceive it as like a sweet, musky floral scent. Exposure, or studies have found that exposure to androstenone can reverse anosmia. So they found that repeated exposure can cause people to be able to smell it after not being able to previously. And this might be caused by an environmental activation of non-functional genes that code for receptors that fit that chemical. Alrighty, so that is all we have for smell. So we're on our last few lectures, so I will be posting the taste lecture soon. And I hope you guys have a great week. I look forward to talking to you guys again. Bye.